1 minute, 59.42 seconds. That is the time it took Eliud Kipchoge to run 42.195 kilometers, a full marathon, on October 12, 2019, in Vienna, Austria. For context, the average recreational runner takes 4.5 hours. Even elite club runners rarely break 3 hours. Kipchoge ran the same distance in under 2. But here is the detail that stops exercise physiologists mid-sentence. His pace, 2 minutes and 50 seconds per kilometer, barely changed from kilometer 1 to kilometer 42. His muscles at the finish line were producing almost exactly the same power output as they were at the start. The explanation you'll hear most often goes like this: Kipchoge has exceptional mental toughness. He has trained harder than anyone. He simply refuses to let his body quit. Coaches say it. Commentators say it. Even Kipchoge himself, in his quiet philosophical way, talks about the mind controlling the body. And that framing, while poetic, is scientifically incomplete, because it skips the actual mechanism entirely. Mental toughness does not stop your muscle fibers from running out of glycogen. Willpower does not prevent hydrogen ion accumulation from disrupting actin-myosin cross-bridge cycling. So the real question is not how tough is Eliud Kipchoge's mind. The real question is, why do his muscles produce a fundamentally different chemical environment than yours when they're under sustained load? The answer lives at the intersection of mitochondrial biology, metabolic substrate efficiency, and a specific form of neuromuscular economy that researchers at the Copenhagen Muscle Research Center, alongside work published in the Journal of Applied Physiology, have been mapping in elite endurance athletes for over 2 decades. What they found is not a story about training volume. It is a story about a molecular infrastructure so specifically adapted that Kipchoge's muscle cells are, in a measurable, quantifiable way, operating on a different energy system than a recreational runner. Not metaphorically different, structurally, chemically, enzymatically different. And that difference begins somewhere most people have never thought to look. Let's start at the most fundamental level. Your muscle fiber, any muscle fiber, yours or Kipchoge's, produces force through a process called the sliding filament mechanism. Actin and myosin proteins interlock, pull, release, and repeat. Each pull cycle consumes one molecule of ATP, adenosine triphosphate, your body's universal energy currency. Think of ATP like a single-use battery. Each contraction snaps one open, extracts its energy, and discards the spent casing, which we call ADP, adenosine diphosphate. The question of muscular fatigue, real molecular muscular fatigue, is almost entirely a question of how fast your cells can rebuild those spent batteries back into usable ATP. Because the moment ATP regeneration falls behind ATP consumption, cross-bridge cycling slows, force output drops, and from the outside, that is what fatigue looks like. A runner whose stride shortens, whose pace slips, whose form begins to collapse. But this is where it gets strange, because when researchers at the Karolinska Institute in Stockholm performed repeated needle biopsies on elite marathon runners during sustained efforts, a procedure where a hollow needle extracts a small sample of muscle tissue while the athlete continues exercising, they found something that upended the simple train more, fatigue less model. The difference wasn't just how much ATP these athletes had available. It was where the ATP was being made and what fuel it was being made from. In elite endurance athletes, the mitochondria, the organelles that perform the most efficient form of ATP regeneration, oxidative phosphorylation, are not only more numerous, they are physically positioned differently, packed in double rows directly alongside the myofibrils, like a dedicated power plant sitting 3 feet from the machine it's running, rather than across town connected by a transmission line. This positional proximity means the ATP diffusion distance, the physical distance a molecule of ATP must travel from where it's made to where it's used, is dramatically shorter. And in biological chemistry, distance is time, and time at the molecular level is the difference between fatigue and endurance. A 2021 study published in the Journal of Physiology by researchers including Dr. Joachim Nielsen found that in world-class cyclists and runners, mitochondrial volume density, the percentage of total muscle cell volume occupied by mitochondria, reached values of up to 12%. In untrained individuals, that figure sits around 3 to 4%. That's not a marginal difference. That is 3 times the aerobic energy infrastructure, packed into the same physical space. Now let's put that infrastructure under stress. Because understanding what doesn't happen in Kipchoge's muscles during a 2-hour effort is more instructive than understanding what does. In a typical recreational runner's muscles, somewhere between 60 and 90 minutes into sustained intense effort, a cascade begins. Muscle glycogen, the stored carbohydrate your cells use as their preferred fast fuel, begins to deplete. As glycogen availability drops, the metabolic process that feeds the mitochondria with pyruvate begins to slow. The mitochondria, starved of substrate, can no longer keep pace with ATP demand. The cell compensates by ramping up anaerobic glycolysis, ATP production that doesn't require oxygen and doesn't require the mitochondria. This pathway is fast, but it's dirty. It produces lactate and critically hydrogen ions. And here's the mechanism of fatigue most people get wrong. For decades, lactic acid was blamed as the villain, the thing burning in your legs, the thing that shuts you down. But research by Dr. Robert Robergs, published in 2004 in the American Journal of Physiology, demonstrated that lactate itself is not the direct cause of fatigue. The real culprit is the hydrogen ions that accumulate alongside it. As intracellular pH drops from a resting level of around 7.1 towards 6.8 and below, it directly inhibits the enzymes that regulate cross-bridge cycling. Phosphofructokinase, one of the key enzymes driving glycolysis, loses activity. The myosin ATPase that powers each contraction slows down. Calcium release from the sarcoplasmic reticulum, the signal that initiates every single muscle contraction, becomes impaired. Here's what nobody tells you. In Kipchoge's muscles during a sub-2-hour marathon effort, this cascade is substantially suppressed, not eliminated, but profoundly slowed. And the reason is threefold. First, those densely packed, adjacently positioned mitochondria are processing pyruvate so efficiently that the demand signal for anaerobic compensation is far weaker. The aerobic system is keeping pace. The shortfall that triggers the hydrogen ion flood is dramatically smaller. Second, Kipchoge runs at a metabolic intensity that sits for him below his lactate threshold, the point at which lactate production begins to exceed the body's ability to clear it. Research by Dr. Andrew Jones at the University of Exeter, who has studied Kipchoge and the Breaking 2 project directly, established that Kipchoge's lactate threshold corresponds to a running velocity significantly faster than his marathon pace. He is, metabolically, running in a zone where his aerobic system dominates almost completely. Third, and this is the detail that generates the most surprise in sports science classrooms, Kipchoge's muscles have an elevated capacity for lactate oxidation within the muscle itself. Type 1 slow-twitch fibers, of which he has an extraordinarily high proportion, contain a protein called MCT1, monocarboxylate transporter 1, which shuttles lactate from working fast twitch fibers into adjacent slow twitch fibers and directly into the mitochondria, where it's converted back into pyruvate and burned as fuel. His muscles are recycling what your muscles are discarding. The net result, measured during physiological testing by the team at Nike's Breaking 2 project in collaboration with researchers from Loughborough University, is that Kipchoge's blood lactate concentration at marathon race pace runs at approximately 2 millimoles per liter. The threshold at which most athletes begin to experience significant performance degradation is 4 millimoles per liter. He races a full marathon at half the metabolic stress level that begins to impair other elite athletes. And this next part is what the data actually shows. Researchers measuring the electromyographic activity of Kipchoge's lower limb muscles during long efforts found something that shouldn't, by conventional models, be possible. His motor unit recruitment pattern, the electrical signal commanding which muscle fibers to activate with each stride, showed almost no drift over extended distances. In most runners, as fatigue accumulates, the nervous system compensates by recruiting larger, less efficient motor units. The pattern becomes messier. More fibers fire to produce the same force. Energy cost per stride increases. In Kipchoge's case, the pattern is essentially the same at kilometer 40 as at kilometer 5. His muscles are not compensating. They are not degrading. They are, by every electrophysiological measure available, doing the same job in the same way throughout the entire event. So how does the biological system reach this state? Kipchoge began running competitively at age 16 under coach Patrick Sang in the Kaptagat training camp in Kenya's Rift Valley, at an altitude of approximately 2,400 meters above sea level. He turned professional at 18. He has now been accumulating specific endurance adaptation for over 25 years. But the mechanisms operating here are not simply the product of volume. They are the product of specific stimuli applied at the right intensity with sufficient recovery over a time frame that allows fundamental structural remodeling. The primary molecular driver of the mitochondrial adaptations we've described is a protein called PGC1-alpha, peroxisome proliferator activated receptor gamma coactivator 1-alpha. It functions as the master switch of mitochondrial biogenesis. When activated by exercise, particularly sustained aerobic exercise performed just below or at the lactate threshold, PGC1-alpha enters the cell nucleus and upregulates the genes responsible for building new mitochondria, increasing oxidative enzyme activity and enhancing fat oxidation capacity. Research from the laboratory of Dr. John Holozzi at Washington University in St. Louis, widely considered the foundational work in exercise-induced mitochondrial adaptation, established that it takes a minimum of 6 to 8 weeks of consistent aerobic training for measurable mitochondrial density increases to occur. Maximum adaptation in elite athletes accumulates over years, not months. Kipchoge runs approximately 200 kilometers per week during peak training, a weekly volume representing roughly 13 to 15 hours of aerobic stimulus. Each session is a targeted PGC1-alpha activation event, repeated hundreds of times per year, compounding over decades. But here's the genetics question, because it always comes up. Here's what nobody tells you about Kenyan runners. Researchers studying runners from the Kalenjin ethnic group, from whom a disproportionate number of the world's greatest distance runners emerge, including Kipchoge, have found genuine structural differences at baseline. A 2011 study published in the Journal of Applied Physiology found that Kalenjin children who had never trained showed significantly higher oxidative enzyme activity in their muscle tissue than age-matched European controls. Additionally, the classic limb proportion study published by researchers at the University of Copenhagen found that Kenyan elite runners have, on average, thinner lower legs relative to body mass, which translates to reduced rotational inertia during the swing phase of running, requiring less muscular energy to cycle each stride. So yes, there is a genetic substrate, but the research is equally clear that this substrate is neither sufficient nor deterministic. It creates a favorable starting architecture. What turns that architecture into the system we've been describing requires the specific, long-term, structured, adaptive stimulus. And this is where it stops being about Kipchoge and starts being about you. What Kipchoge's biology illustrates is a principle that exercise physiologists call the principle of specific adaptation to imposed demands, SAID, but the mechanism beneath it is more precisely described by what researchers term the stress adaptation supercompensation cycle. State it as a law. Biological systems under repeated specific recoverable stress do not merely return to baseline, they reorganize at a structurally higher level of efficiency. This is not a motivational statement. It is a description of a molecular process. The stress of sustained aerobic exercise depletes glycogen, elevates AMP to ATP ratios, and activates the AMPK pathway, AMP activated protein kinase, which in turn triggers PGC1-alpha, which triggers mitochondrial biogenesis, which increases oxidative capacity, which raises the lactate threshold, which pushes the fatigue cascade further down the timeline. The same principle operates with different molecular machinery in completely different biological contexts. In neurons, repeated activation of a neural pathway produces long-term potentiation. The synaptic connections physically strengthen, reducing the signal threshold required to fire. This is why a tennis player's reaction to a fast serve becomes automatic. The neural circuit remodels. In bone, repeated mechanical loading activates osteoblasts, bone-forming cells, through a mechanotransduction pathway involving piezoelectric signaling in the bone matrix. The trabecular structure of the bone reorients along lines of stress. The bone becomes specifically stronger in the directions it is repeatedly loaded. In every case, the principle is identical. The system reads the repeated demand as a survival signal. It rebuilds not just to handle that demand, but to handle it more efficiently next time. Kipchoge's muscles are the product of 25 years of sending exactly the right molecular signal, at exactly the right intensity, with exactly the right recovery interval, allowing the system to rebuild, not back to baseline, but slightly above it, hundreds of times over. The result is a biological infrastructure that is, by any molecular measure available, 3 to 4 times more aerobically capable than the system you were born with, or that a recreational runner maintains. So now, when you go for a run and you feel that burn arriving in your legs somewhere around the 20-minute mark, the tightening, the heaviness, the sense that your pace is becoming unsustainable, you know what's actually happening. It is not weakness. It is not poor fitness in any general sense. It is a specific molecular event. Your mitochondrial density, your lactate clearance capacity, and your oxidative enzyme concentrations have reached their current ceiling, and your anaerobic system is being asked to compensate for the shortfall. That feeling is not your body failing. It is your body telling you exactly where its current biological ceiling is. And now you understand that that ceiling is not fixed. Every sustained aerobic effort you complete sends an AMPK signal to your muscle cells. Every signal triggers a small increment of PGC1 alpha activity. Every PGC1 alpha activation contributes over weeks and months to a measurable increase in mitochondrial density. You will not reach Kipchoge's mitochondrial volume density of 12%. But going from 3.5% to 5%, a shift well within the documented response range for recreational runners following structured aerobic training, means the burn arrives later, is less severe, and clears faster. The science is not showing you a gap that cannot be closed. It is showing you the precise molecular handles that, when pulled consistently and intelligently, move the curve. Kipchoge did not start with the biology we've described today. He built it, one PGC1 alpha activation at a time, over 25 years. The difference between him and every other runner on Earth is not, at its core, a mystery. It is a measurable, well-understood biological infrastructure built by a specific process, governed by specific molecular rules that your cells follow just as faithfully as his. But here's the question the data raises that we haven't answered yet. If the molecular mechanism of adaptation is the same in every human muscle cell, the same AMPK pathway, the same PGC1 alpha signal, the same mitochondrial biogenesis process, then why do some people, even with equivalent training, plateau while others keep adapting? Why do two runners with identical weekly mileage, identical intensity distribution, and identical recovery protocols produce measurably different mitochondrial adaptations after 3 years? The answer involves a mechanism that is, in many ways, more surprising than anything we've covered today. It involves not what's inside your muscle cells, but what's regulating the expression of the genes that build them. A field called epigenetics, and specifically a process that researchers at the Karolinska Institute have now linked directly to the inter-individual variation in endurance adaptation. That's the subject of the next video. If you want to understand why your genetics are not your ceiling, and what is actually determining how high your ceiling goes, that video is where to go next. Subscribe to the Performance Lab, not for motivation, not for training tips, but because once you understand the biology, you see the mechanism, and once you see the mechanism, you cannot unsee it. The next video is already waiting for you.